Assalamualaikum. Hi everyone. Welcome back to my channel. I am Muhammad Shahril Amirul bin Muhammad Jafar, and today we will be discussing practical 2 from our ecology course, focusing on the factors that affect the distribution and abundance of organisms. Let's dive in.
Let's meet the team member who contributed to this project. The first one is Ahmad Nazli Shah bin Muhammad Nazri. Next, Ahmad Fauzan bin Kamar Zaman and Muhammad Shahril Amirul bin Muhammad Jafar. In this practical, will be reviewed by Dr. Nutabita binti Sheikh Nasir as our lab advisor. Introduction. All of the organisms that exist in an ecosystem are its biotic components. Population is defined as an organism of the same species found in the same habitat. Communities are made up of populations of several species living together and interacting in the same area. One method for analyzing the interaction of organisms in a community is to inventory all of the species in the area and compare their population levels. We will be introduced to the basic idea of a quadrant in this practical. In addition, we must categorize the plant species found there and monitor the abiotic elements of the study site.
Our objective for the practical are to introduce the simple concept of quadrant. Number 2, to enhance the power of observation. Number 3, to record the difference in plant distribution within the quadrant. Number 4, to infer from natural observations the possible factor that affect distribution and abundance of organisms. For this practical, you will need raffia string, scissor, measurement tape, notebook, and a stick.
Methodology. Number 1, our practical study focus on exploring different areas of forest at UiTM Jengka, Pahang. Number 2, one quadrant of 5 meter by 5 meter at each forest type were made. Multiple quadrant samples were performed throughout the habitat at several random locations. Number 3, all tree stands that are found inside our quadrant were recorded and observed. The data was used to estimate the population size and population density within the entire habitat. Note, the area to conduct our quadrant was selected which had a wide variety of plants.
Our results showed significant variation in the number of species across different quadrants. We identified 34 species, with species Y having the highest individual count, indicating that the environmental conditions at the site were particularly suitable for its growth. This is the result for quadrant 1. This is the result for quadrant 2. This is the result for quadrant 3. This is the result for quadrant 4. And this is the result for quadrant 5.
Based on the result, we can discuss that the quadrant sampling method is a straightforward way to study plant distribution and abundance. It's important to choose the best place for sampling because careful observation within the quadrant reveals patterns in how different plant species are distributed. A 5 meter by 5 meter quadrant is ideal for studying herbaceous plants, small shrubs, and sedentary organisms like ants. Areas with direct sunlight will have different plant species compared to shaded areas. Factors like light availability, soil's property, nutrient levels, and water availability, along with biotic interaction such as competition and facilitation, influence plants community composition and species distribution. Choosing the right quadrant size is crucial. Too small a quadrant might miss many individuals. underestimating population size. Too large a quadrant can introduce errors and be impractical for throughout for true survey.
Our results show 34 species, with species Y being the most abundant. The environmental conditions at our site favored species Y due to suitable abiotics and biotic factors like soil, water, and sunlight. Some species, such as E, V, W, S, AE, AF, AG, and AH, were found only once, indicating they may not thrive in that site. These species were spread across three quadrants, showing their distribution is sparse. Spatial heterogeneity, where plants compete for nutrients, also affect plant distribution. Factors influencing plant distribution. First, abiotic factors such as soil properties, water availability, light availability. Second, abiotic factors such as competition, facilitation, and herbivory and predation. Third, time since disturbance.
Based on this practical, we can conclude that the simple concept of quadrat has been successfully introduced. Quadrat sampling does help in understanding and determining factors that affect the distribution and abundance of organisms. The power of observation has been successfully enhanced. From this practical, the distribution and abundance of organisms are influenced by lots of factors like abiotic condition and biotic interactions. Difference in plants distribution within the quadrat has been successfully recorded as shown in the table results. It indicates that the plants are variety in species and or populations. The possible factor that affect distribution and abundance of organisms has been successfully inferred from natural observation. This practical emphasizes the complex interplay of abiotic and biotic factors in shaping the distribution and abundance of organisms within ecosystem.
To wrap up, here are some post-lab questions to reinforce what we have learned. Number 1. Define the term quadrate in practice. Number 2. What are the two basic requirements of all quadrate techniques? Number 3. Discuss the advantages and disadvantages of using larger or smaller quadrates. Number 4. Explain the issues concerned with ecologists in conducting sampling methods in complex habitats. And the last one, hypothesize the factors that would cause variations in plant species numbers within quadrates.
Thank you for joining us in this exploration of ecological factors affecting plant distribution and abundance. If you have any questions or talks, please leave them in the comments below.
Don't forget to like, share, and subscribe for more educational content. Until next time, keep exploring the wonders of science.
Bye!